continue when you want.
Okay. I wish to start by sincerely thanking the organizers of this international conference of psycholinguistic and neurolinguistic research, and particularly Professor Olga Ivanova for kindly inviting me to present this plenary talk during this first session of the conference. I would also like to thank all the people in the virtual room for being here. In my talk, I will discuss fresh experimental and theoretical approaches to the nature of language disorders that aim to integrate recent discoveries about their biological nature in the domains of genetics, brain physiology, or behavior. Hopefully, these approaches should help physicians, speech therapists, and clinical linguists to better interpret their findings, improve their understanding of the etiology and symptomatology of disorders, and ultimately achieve earlier and more confident diagnosis as well as more efficient therapy. More specifically, I will argue in favor of a classification of language disorders based on abnormal patterns of brain oscillations during language processing, because brain rhythms are intermediate level biological components of language in between molecular factors at the bottom and language features at the surface. They are expected to provide more biological motivated and more theoretically grounded accounts of disorders, and particularly robust endophenotypes that can be used as reliable hallmarks for achieving earlier and more accurate diagnosis. On paper, clinical categories like dyslexia or specific language impairment refer to cognitive disorders in which only language is impaired and that can be differentiated, cognitive, neurobiological, and genetic, and more difficult to handle, essentially because at all those levels. This circumstance is expected to impact negatively on the diagnosis and the therapeutic approaches aimed to ameliorate the symptoms and deficits associated with these conditions. To begin with, patients commonly show symptoms that are compatible with more than one disorder, linguistic or not linguistic by nature, to the extent that comorbidity is frequently observed during clinical practice. At the same time, people suffering from a particular disorder usually exhibit linguistic disabilities, but also abilities that are pretty variable. In order to apprehend this variability, different subtypes of the same disorder are usually coined, in which one aspect of language is claimed to be more impaired than others. Importantly, problems with language at the surface, so to speak, are only indirectly related to the cognitive deficits at the bottom. This similarly contributes to increase the variability of the symptoms and makes the categorization of disorders more troublesome. On the whole, it seems that different disorders, or even different subtypes of the same disorders, can emerge from the same cognitive deficit. Hence, the alleged heterogeneity and or comorbidity. In other circumstances, however, different deficits that can be specific or not specific to language can contribute to the same disorder if they converge on a similar symptomatic profile. It is then possible that in each subtype of a particular disorder, a different deficit prevails. Hence, the reported heterogeneity and comorbidity too. Another important concern is that very frequently problems with language concern to quite broad aspects to the extent that they do not match components, features, or computations that are important for modern linguistic theory. As a consequence, clinical typologies are sometimes weird for linguists For instance, according to some accounts of a specific language impairment, there exists a syntactic pragmatic subtype of this condition. However, linguists carefully differentiate between the knowledge of a language, that is the grammar, and how this knowledge is put into use, that is pragmatics. Actually, for most linguists, and for most neurolinguists indeed, pragmatics involves many other things besides our linguistic competence. Finally, consider that the clinical profile of patients usually change throughout development to the extent that they can even switch from one subtype of the disorder to another subtype during the course of their lives. Accordingly, one cannot simply assume that the deficit observed in the adult state are the same as those apparent in the infant state, and vice versa. Things are quite similar at the neurobiological level. Accordingly, the brain areas found affected in one disorder can be found impaired in people suffering from a different disorder. Moreover, it's frequently observed that the affected regions give rise to mixed symptoms. Overall, it's not clear whether the involved regions are multifunctional by nature or perform instead some basic computations that are required for language and for other cognitive abilities. Lastly, it commonly happens that the exact localization of the regions of interest for a particular disorder change from one affected subject to another. Things are not easy to interpret at the molecular level either. Many candidate genes and risk factors for different language disorders have been identified today. However, it is not one but many genes that usually contribute to each disorder. Typically, several pathogenic variants of each candidate gene can be identified that contribute to the language abilities of the neurotypical population. What is more, the same mutation in the same gene can give rise to different disorders in different subjects to the extent that candidate genes for a particular disorder are frequently mentioned as candidates for several other clinical conditions. Conversely, pathogenic variants of a particular candidate gene are frequently absent in people affected by the disorder. Finally, it is frequently observed that mutation in genes encoding proteins that are functionally related, if, for instance, one regulates the expression of the gene encoding the other, can give rise to different disorders in different subjects. In order to illustrate this messy situation, I will use dyslexia as a leading example. As it is well known, people suffering from dyslexia have difficulties for properly reading text and spelling words. Notice, however, that reading difficulties are observed in many other cognitive disorders, and not surprisingly then, dyslexia can be comorbid with language disorders like specific language impairment or speech sound disorder, but also with attention deficit hyperactivity disorder. Of course, these other disorders can be in turn comorbid with other different conditions. Dyslexia has been hypothesized to result from the dysfunction of the phonological component of the working memory. However, this deficit also causes specific language impairment and speech sound disorder. Conversely, several other deficits have been claimed to contribute to dyslexia, including problems with categorical perception, problems for processing and discriminating short acoustic pulse, cerebral dysfunctions, difficulties with visual processing, or the dysfunction of the magnocellular pathway. Of course, these deficits can result in other different conditions, many of which are comorbid with dyslexia. Regarding the brain anomalies observed in dyslexics, consider, for instance, the ventral portion of the occipital temporal region. This area supports in part our reading skills and is found underactive in dyslexics during reading tasks. However, it seems to be involved as well in the recognition of faces, and it has been related to prosopagnosia too. Finally, several candidate genes for dyslexia has been identified today, most of which regulate axonal growth and neuronal migration in the cortex, plausibly accounting for the structural and functional anomalies observed in the brains of people with this condition. However, as you can see in the slide, these genes are just for language or even cognitive disease. As noted, candidate genes for a particular condition can be functionally related to genes resulting in another different condition when mutated. Hence, the dyslexia candidate ROBO1 interacts with a gene called FLNA, which is a candidate for brain heterotopia. In turn, the FLNA protein binds SMIP, a candidate for a specific language impairment, as well as FOXP2, the famous language gene, which is a candidate as well for a specific language impairment, but also for dysarthria. Now, FOXP2 targets CNTNAP2, and mutations in this gene result in a specific language impairment, but also in stuttering, language and mental delay, and autism. The advent of the so-called omics revolution is more complex. On the one hand, the amount of biological data about disorders has grown exponentially. On the other hand, we have learned that there are additional levels of complexity that need to be explored if we really want to gain an accurate picture of the real nature of language disorder. Accordingly, epigenetic changes, modifications in protein networks, alterations of signaling pathways, abnormal patterns of neural assembly, or aberrant patterns of neural synchronization need to be considered on a part to gene mutation, abnormal neuroimaging results, or language deficits. Importantly, as I will try to show later when discussing about brain oscillations, it might well be that the specificity of language in cognition, and hence the idiosyncrasy of language disorders that we were unable to find at the genetic or the symptomatic levels, can be found instead at some of these other levels of biological complexity. Overall, it seems that clinical linguistics, which has traditionally focused on the cognitive and linguistic evaluation of patients, and in the analysis of corpora of disordered language, face today a triple challenge. First, it needs to integrate new types of evidence as provided by many different areas of research interested in language disorders. I will call this the challenge of multidisciplinarity. Second, it needs to incorporate new methodologies and tools like next generation genetic tools, which are often hard to understand and use. I will call this the challenge of technification. Third, it needs to formulate new hypotheses about the etiology of language disorders that take into account this increasing body of evidence, and particularly fresh models of development and evolution as currently discussed in the domain of biology. I will call this the challenge of theorization. I believe that this is possible if a system biology approach to language disorder is adopted. One approach that it is aimed to characterize from a holistic perspective the complex interactions among the myriads of biological components involved, as well as the emergent properties resulting from such interactions. Among other things, this approach can be expected to circumvent the limitations of current typologies of language disorders, which mostly rely either on symptomatic criteria or on etiological criteria. Typologies based on symptoms often fail to categorize and characterize patients unambiguously, essentially because of the widespread problems of comorbidity and heterogeneity, as discussed earlier. Also, they usually fail to incorporate etiological factors in a disorder-specific way, particularly genetic and neurobiological causes of disorders. For instance, as far as genes are concerned, we know now that language disorders do not result from the alteration of a small number of disorder-specific genes, but of many, if not most, of the genes present in the brain. Accordingly, it might be better to characterize disorders in terms of whole brain disorder-specific transcriptomic profiles. At the same time, etiological classification of language disorders often fail to explain why different disorders can result from the alteration of the same molecular components. Here, two opposite symptomatic profiles, namely schizophrenia and autism, result from mutations in genes involved in the same biological process, namely neuronal synapses. Actually, this messy scenario, as I have called it earlier, can be easier to interpret not just if we adopt a systems biology approach to language disorders, but more specifically, if we rely on eco-evo-devo theories in biology, which build on the deep link that exists between the environment, development, and evolution. According to this approach, language can be claimed to have evolved as a result of specific changes in the developmental path of the hominin brain in response to change in the environment in which our ancestors lived. And it is the most recently evolved components of human cognition that are expected to be the most sensitive to the deleterious effect of developmental perturbations resulting from environmental changes, either internal to the organism or external to it. This circumstance similarly explains two generalized findings of clinical linguistics. First, although as noted, the etiology of disorders is quite diverse, some deficits are shared by most conditions, and they usually concern to phonology, morphology, and to demanding computational processes like agreement. Second, although as also noted, the number of etiological factors is big, the number of disorders is far smaller. Under this eco-evo-devo view, these two findings are not surprising. Regarding the first one, language evolution has been also viewed as the emergence of new interfaces between basic cognitive blocks that are otherwise very robust after millions of years of stabilizing selection, and that as a consequence are not usually found impaired in disorders. By contrast, because of their evolutionary novelty and because the reduced resilience of the neural networks supporting them, these new interfaces are more sensitive to damage. Regarding the second one, that is, why the number of disorders is far smaller than the number of etiological factors involved, it can be hypothesized that the disorders finally observed are the only possible phenotypes resulting from the interaction of the factors, normal or abnormal, that regulate brain development. In eco-evo-devo theories, the limited set of phenotypes available during development are construed as specific areas within a morphospace or adaptive landscape. Putting it differently, developmental dynamics canalizes development towards a limited number of ontogenetic paths. Accordingly, what we call the neurotypical language-ready brain can be viewed as the outcome of a successful canalization of the otherwise pervasive developmental noise, that is, gene mutations, minor brain anomalies, and the like. By contrast, what we call language disorders can be viewed as the outcome of a suboptimal canalization of more severe brain mutations, severe brain damage, and the like. What we need now is to determine which are the best parameters defining the language morphospace or the language adaptive landscape. Consider as a good example the shells of ammonites and nautili. Similarly to language disorders, the number of different shell forms was small: coil, uncoil, and helical. Nonetheless, shell morphology depends on two parameters: reed suspension rate and reed coiling tightness, which changes continuously, similarly to what happens with the etiological factors of disorders. As far as language disorders are concerned, one interesting possibility could be that, as noted, relying on aberrant gene expression profiles in the brain. Accordingly, whereas some expression profiles would be characteristic of different language disorders, other, or perhaps others, would be associated to the neurotypical language-ready brain. Nonetheless, in our past research, we aim to check whether brain rhythms could be a better or perhaps the best candidate for properly defining the morphospace of language development in the species, either pathological or neurotypical. Several reasons justified our interest in brain oscillations. First, they are primitive components of brain function. Second, we can expect them to be connected to some computational primitives of language, thus allowing to understand and not just to localize brain functions. As famously pointed out by David Popel, past neurolinguistic studies suffer from two crucial limitations. On the one hand, they usually made two broad conceptual distinctions: syntax versus semantics, morphology versus syntax, and the like, which actually involve multiple and diverse neural computations and representations. On the other hand, the fundamental elements of linguistic theory cannot be reduced or matched up with the fundamental biological units identified by neuroscience. Quoting Poepel, it is then urgent to spell language in computational terms that are at the appropriate level of abstraction, that is, that can be performed by a specific neuronal population. Many people now regard brain rhythmicity as one and perhaps the most suitable of these appropriate levels of abstraction. For example, the assignment of language-relevant features like tense or case can be satisfactorily interpreted as the embedding of high-frequency oscillations inside oscillations operating at slower frequency. Similarly, some rhythmic features of speech have been successfully related to specific brain oscillations. A third reason is that the hierarchy of brain oscillations has remained remarkably preserved within mammals during evolution. Not surprisingly, the human-specific pattern of brain activity supporting language can be evolutionarily linked to the oscillatory signature of the primate brain. As my colleague and co-author Elliot Murphy discusses in a recent paper, the emergence of the human language oscillome, that is, the language-related phasal and cross-frequency coupling properties of neural oscillations, seemingly reshaped the ancient primate oscillome we inherit. At the same time, the reverse also happened, as this is why we find in human languages that semantics impose particular constraints on possible syntactic structures. A final but very important reason is that there is evidence that each cognitive disorder distinctive profiles of brain rhythmicity. This is of particular interest for clinical linguistics because brain rhythms are highly quantifiable and heritable traits. They might result in robust biomarkers or endophenotypes of language disorders. For example, a gamma band dysfunction has been repeatedly hypothesized to be a robust biomarker of autism spectrum disorders. Nonetheless, if we really want to use these oscillopathic profiles as reliable biomarkers or endophenotypes of language disorders, we first need to map these abnormal disorder-specific patterns of brain oscillation to the language deficits that are also typical of each condition. Pretty obviously, this translational effort should also allow to gain a better understanding of the etiology of these disorders, particularly of the effect of gene mutation. In our past work, we found evidence that this translational effort is possible. For example, degraded gamma and theta synergy may explain the problems that people with autism experience with speech perception, tone recognition, and parsing phonemic representations. The reason is that when processing speech, gamma oscillations correspond to phonetic features and are important as well for accessing to stored templates. Specifically, in our research, we have shown that it is possible to draw bridging links between candidate genes for conditions like autism, schizophrenia, or dyslexia, abnormal disorder-specific brain oscillations, and language problems. For example, the gene ZNF804A encodes a zinc finger binding protein, and it is highly expressed in the hippocampus and the neocortex, particularly during late embryonic development. The hippocampus is a source of theta bands, which play a major role in coordinating distributed cross-cortical activity, in particular activity in the prefrontal cortex. Because of their involvement in working memory as a filter that imposes memory-related rules, and because of the overall role of the hippocampus in the transformation of individual experience into semantic structures such as maps and schemas, hippocampal theta is expected to explain core aspects of language processing, both syntactic, like the chunking of syntactic objects, and semantic, like category fluency. Pathogenic polymorphism of this gene have been related to semantic problems in subjects with schizophrenia. Accordingly, significant decreases in the coactivation of the right hippocampus within the whole hippocampal network, as well as in intrahippocampal theta band, have been found in risk homozygotes for one variant of the gene. Interestingly, people with this risk allele show a greater coactivation of the hippocampus and the prefrontal cortex, specifically the superior frontal gyrus. Likewise, several polymorphisms of this gene have been associated to verbal deficit in people with autism, who exhibit a reduced expression of the gene in several brain areas, like the anterior cingulate gyrus. Just to put another example, green 2A is a gene that encodes the subunit 2A of the MMDA receptor, which plays a key role in long-term potentiation, important for memory formation and consolidation. This function seemingly results in part from its regulation of gamma oscillation formation and modulation. Mutations in green 2A are found in people suffering from different forms of epilepsy aphasias, like Rolandic epilepsies or Landau-Kleffner syndrome. Specifically, they are associated with errors in articulation and with problems with pitch and prosody, which pertain to the syntax-phonology interface. In turn, these deficits can be tracked to an abnormal gamma activity involved in the processing of fast-rate phonemic and syllabic information. Interestingly, most of the candidates for language disorders in conditions like autism, schizophrenia, or dyslexia that play a role in brain rhythmicity are functionally interrelated. Moreover, they map on particular regulatory pathways, cell types, or functions, as well as aspects of brain development and function of relevance for language processing, particularly through dopaminergic, GABAergic, and glutamatergic synapses. Importantly, too, these genes exhibit a distinctive disorder-specific pattern of up and down regulation in the brain of patients, as shown here by the expression grids of these genes across the brain regions profiled by the Allen brain atlas. Notice that the specificity of the molecular signature of each disorder relies not on the set of genes involved, which are essentially the same, but on their expression patterns in each brain region, which is different in each condition. It is important to know that several of these genes that contribute to basic aspects of the oscillatory activity of the language-real brain show differences in their methylation status between Neanderthals and humans. Pretty obviously, we cannot track the oscillatory activity of the brain of extinct hominins, but as shown before, from molecular differences that are suggestive of differences in the expression pattern of the genes, we can infer differences in cognitive function important for language through potential changes in brain rhythmicity. In summary, because of their bridging role between genes at the bottom and language features at the surface, both developmentally and evolutionarily, brain rhythms could eventually be the biological level at which the specificity of the language problems featuring each disorder emerge. Certainly, this research has an applied dimension too. If we succeed in this translation of language dysfunction into disorder-specific patterns of brain abnormal oscillations, we might be able to diagnose language disorder earlier and in more accurate ways. In my opinion, several complementary lines of research would be very welcome for improving this approach to language disorder that heavily builds on brain rhythms. First, we need to disentangle the molecular mechanism that channel and fail to channel variation at all levels of biological complexity. Second, we need to improve eco-Evo-Devo-friendly depictions of the modularization of the disordered brain. Third, we need to optimize our current models of the language ontogeny in people with disorders. Finally, we need to pay attention to emergent properties of language and to properties that fail to emerge, because language is undoubtedly a complex system, and many of the properties of complex systems are emergent by nature. Summarizing what I have argued today, I think that a characterization of language disorders based on intermediate level components, and particularly on the coupling between brain oscillations during language processing, is more biologically motivated and more theoretically grounded than others, particularly than most of the typologies currently used by clinical linguists, either based on symptoms or on causes. Accordingly, this fresh characterization is expected to help us to identify more robust endophenotypes of language disorders that can be used as reliable hallmarks from an earlier and more accurate diagnosis of these conditions. Well, thank you very much for your attention.
Okay, thank you very much, Antonio, for such an, as usual, insightful lecture. So we have 20 minutes approximately for comments and questions. So you're really welcome. You can switch on your microphones to ask questions or pose them either through chat. Anybody? Anshu, do you want to ask any question? Yes? No questions?
I have a question.
Yes, of course. Yes, Jordi, please.
Hi. Cool. Hi, Antonio. Nice talk. So you were focusing on developmental disorders, right? So I wonder to what extent would you also extend this oscillatory account to acquired disorders or impairments like aphasia, for example, that have, let's say, a more grammatical focus than dyslexia, for example?
Yeah, thanks for your question. I think that this can be also applied to acquired language disorders, but I am more concerned with genes. So this is why I have focused on developmental language disorder, particularly because we are trying to find some specific change that might account for difference with, extinct hominids. But I know that many people is working with acquired disorders from this perspective. Yes, there is plenty evidence that this condition also involves an abnormal brain rhythmicity that can be related to the language deficit observed in patients. So there is room for research in this area too, of course.
Thank you.
Hello. Hello.
So, yes,
Yes, please.
Can I ask?
Sure.
Sir, is there any standard to measure the functional MRI for this aphasic or language disorder patient, autism cases?
You mean if there is fMRI studies about these conditions?
For fMRI, is there any standards? Some standards to how you can measure those in autism and in aphasia?
Yeah, I'm not an expert because I mean I do not do this research. I just collect data for other studies, but I know that there is some attempts to provide some kind of clinical guide or some some map in order to try to diagnose this kind of disorder in advance. Well, this is actually the aim of this approach based in oscillations, because what you are trying is to find these abnormal patterns of brain activity well before the emergence of first symptoms of disease. So you can try with some strategies to ameliorate the symptoms, or even to prevent or delay the emergence of these symptoms. So there is many people trying to find early diagnostic tools of these disorders, and
yes,
sure, these fMRI maps should be really helpful, as well as, for instance, a change in the expression of candidate genes in the blood. It's also, there are many people working on these abnormal profiles of gene expression in the blood of people with autism, for instance.
sure. Thank you. Thank you very much.
Okay, is there any other question? Anybody wants to ask anything? No?
May I?
Yes, of course.
I
Katerina, yes,
raised my
you're welcome.
hand. Thank you very much. Thank you very much for your talk. It was really inspiring for me. I was wondering if we can make any hypothesis regarding the onset of problems in brain activation, depending on different diseases. Is there any?
Oh, could you please repeat your question, because my connection had some problems.
Okay.
I couldn't hear you. Sorry.
I was wondering if we can make any hypothesis regarding the onset of problems in brain activation, in dysfunctioning in brain rhythms based on
Well,
oscillation.
yeah. Yeah, it's difficult, because you know that even if this disorders has a genetic basis, the emergence or the onset depends on environmental conditions too. So there is a complex interaction between genes and environment. So it is quite difficult to predict the onset of these conditions. But sure, if you could track, in some sense, change in, for instance, the expression patterns of genes, not in the brain, of course, but in the blood, you may perhaps predict the onset of some, if not the whole condition, but some symptoms of the disease. But the problem is that this is not, at present, this is not very accurate because of this complex interaction with environmental factors. So this is why I highlighted whole brain expression profiles, for instance, as perhaps more useful tools, because they consider all the genes expressed in the brain according to this recent omnigenic hypothesis about complex conditions that are with that conditions actually does not result from the mutation of one or two genes with high penetrance of strong effect on the brain, but from subtle change in the expression level of most of the genes expressed in the brain. So this is another important step towards an early diagnostic, characterization of diagnosis of these conditions. But yes, this is quite preliminary, and we need to spell out all these complex interaction across all the levels of complexity I highlight during my talk.
Thanks for your answer.
You're welcome.
Okay, any other question? We may have some 3 minutes probably if somebody wants to ask anything. No? Okay, well, Antonio, thank you very much for your presentation. I stop recording here, if I can.